he's going to be taking a faculty position. This is actually, I'll keep talking while he's getting things set up. He's, this is his second visit here to MSU in the last couple of years. He gave a talk as part of the neuroscience program some time ago. And so as things get underway, we'll just turn it over to Daniel.
Probably should have done this during the questions. Let's see. Oh wow. It's been all day here. Can everybody hear me okay? Yeah, that sounds good. So in the spirit of the new frontiers, I thought I would give my talk a rather ambitious title, which I could get away with because my title is not on the program. At the same time, I thought that I should emphasize that this is a rather simple animal. So, omni-neuronal recording from tiny brains. I'm very impressed with what the previous speaker told us about how much they can achieve in just this small amount of, well, relatively small amount of software. And I think it is very important for the next generation of people like Jeff Hawkins to have continued insight of what actual brains do. And I just wanted to give you one example of what our brains do. Unfortunately, I lost my speaker view here, which is not going to help me. Anyway, we can integrate information across a host of sensory modalities. Now, this is an example that all of you have probably come across, and I have cycled by this particular one about 200 times before I even noticed that there was something odd going on here. None of us have any problem behaving appropriately in this sort of situation. But imagine that you are William Shakespeare. You would recognize that, have no problem with that. But this wouldn't mean anything to you, probably. Or imagine that you grew up in Greece and you haven't learned English yet. Then, oops. Then you'll be able to recognize that, but that might not mean anything to you. Healthy adults have no problem with this at all. The ability to do this, though, is compromised in several neuronal diseases. For instance, schizophrenics might have problems integrating this sort of sensory cue, and they might be very upset by it. People with certain kinds of autism spectrum disorder might be very confused by this sort of thing. And your brain is not just integrating these cues, it's also seeing, wow, it's a very nice day today. I'm sad that I'm going to have to be in lab. Or it's good that it's a little bit cooler today. Or, whoa, that's an ambulance in the back. I better actually get off the road. You know, your brain is doing a whole lot of things at once. So that's just one example of what a complicated brain can do and what I would really like to understand. Now, the human brain, as we saw, is a rather complex beast. We have the neocortex with about 100 billion neurons, and then all the stuff in between that makes us a functioning animal. And of course, there are about 1,000 connections between that any neuron gets from its friends. We would like to understand this system, but it's rather difficult to record comprehensively from such a large system. Plus, there are rather few volunteers for recording. Does anybody? I was assuming that I was going to have this presenter view here. Does somebody have a clock that I can use? Because I realize I don't think I have my. Aha! I'm in a position of power. That's a really tricky. You know what? I'm going to spend I'm going to spend 1 second of your time to seeing if I can connect this to the monitor so that it actually understands that there are two screens here. I don't think that's going to happen quickly enough. It's not good enough at Windows, sorry about that. So the point here is that there are an awful lot of neurons in the brain. In fact, there are about as many neurons in your brain as there are stars in the galaxy. Of course, these stars in the galaxy pretty much interact only by gravity, which is rather simple. And in contrast, your brain connects, all those cells connect in many, many ways in very complicated temporal patterns. So this is hard. And neuroscientists would love to understand what's going on here, but I think many of us think that it's going to be more than our lifetime to do this. So what do we do? Well, one thing that you can do is study a mouse instead. That gains you a factor 1,000 in sheer neuron count and something similar in connection count. That's still a pretty big number. That's about the same number of neurons that you have bytes in the computer right now. You could go to a fruit fly and get another factor 1,000. Now we're getting more tractable. Or you can go to my friend the leech. Now the leech has 10 to the 4th neurons, so that's not very different from the fruit fly. But you gain one advantage. It's such a primitive system that it is very strongly segmented. And in fact, each of these little ganglia that you see along its body is pretty much an identical copy of all the other ones. And so the functional system here has only about 400 neurons, which is a relatively tractable number. Plus, of course, if we work on the leech, we don't need to ask for volunteers. So what are we going to do? We want to study the neural basis of behavior. Here is a schematic of the leech nervous system with one of these ganglia shown in great detail. Now the beauty of the leech, and here is what it actually looks like. This is one of these ganglia, and here is the skin of the leech. That's about 0.5 millimeter across. This is about 1 centimeter. The beauty of this system is that all of these cells are in predictable or reliable locations. So if you open one leech, you will find that there is this nociceptive cell right over here. It might be shifted a little bit, but it's going to be roughly there. And that same cell you'll be able to find in all of these ganglia in any leech. So that's an electrophysiologist's dream. You can actually reliably record from most of the parts in this system. Now, still, this is a real animal. It can do lots of things. It can swim, it can crawl, it can find prey, it can bite, it can eat, it can mate. All of those things it can do, and it does that using these very few cells. So we have an opportunity here to record from a much smaller cell ensemble than you could even in the fruit fly, and understand the complete behavioral output of the mind. And in fact, this has been done to some degree in various systems, and I'm going to talk to you about those. So my grand aim is to figure out all of the leech sensory processing systems. And to end up with this schematic similar to what Feldman and Van Essen did in the early 90s for the visual system of the macaque monkey. Now, this is an amazing body of work. They created a connection diagram in the monkey cortex of all the visual areas and created a connection diagram much like you would show of an electronic circuit diagram. There's a big caveat here though. Each of these little boxes does not represent an individual neuron. It represents a whole big area that has millions of neurons in it in some cases. And it doesn't tell you in any way how you could reconstruct this. Because all of these boxes are black boxes. And each of these lines here represent millions of connections. Now in the leech, life is much simpler. Here is a complete circuit diagram, as far as we know, of swimming. And I'm not hiding anything. I'm just showing you 10 or so neurons that are the central pattern generator for swimming in one ganglion, and 10 or so neurons that are the central pattern generator for swimming in the posterior neuron, as we knew it in 2001. These connections here are each individual synaptic connections between these cells. And we thought, at least at this point, that this was more or less complete. So what we can do in principle here is at the single neuron level create a connection diagram and understand functionally what's going on in the full behavior at much greater detail than you would ever hope to achieve in a manual. Now, this work was done by sticking electrodes into cells one at a time or a pair at a time. But even if you have only 400 neurons, there are 16,000 pairs, and so that gets rather tiresome. So what we would like to do is to record from all of these cells at once. And one very nice way to do that is using voltage-sensitive dyes. And I'm going to pretend that I have time to explain that to you. The cell membrane can exist basically in two states. It can be hyperpolarized, the cell is at rest, which is the normal state, or it can be depolarized, the cell is active. And that means that there is going to be a voltage across the cell membrane here. So what we do is we stick a big molecule in the outer leaf of the cell membrane that is fluorescently active. If you shine ultraviolet light on it, it will emit in blue. We also set a second little molecule that can travel through the membrane. This molecule doesn't do anything when the cell is active in this case because it's negatively charged and it sits here far away on the inside of the cell. But when the cell goes to its resting state, that little molecule drifts upwards and now some magic occurs. Some of the energy that the ultraviolet light puts into the big molecule gets transferred to the little molecule, and the little molecule now emits that light at a different wavelength. And so if we use a pair of cameras to look at the cells, and we image both the blue light and the green light in this example, we can look at individual cells and see whether they're active or not. And this actually works. So here's an example of what that looks like during swimming. And I'm going to see if I can give you some sound of this, because it's much more fun if you can hear it. Okay, so what you saw there was that all of these cells, which is about 1/4 of the cells in the nervous system of this one ganglion, are recorded, and they get brighter and dimmer over time. Many of them actually synchronize with the swimming rhythm that you could hear from one of the nerves that go out to the musculature. I'm going to let you listen to that again. I am severely technologically challenged today, I'm sorry about that. My mouse cursor disappeared. So what you would see, if I were able to show you this movie again, is that many of the cells participate, either get active or inactivated during those bursts, and there are some cells that have nothing to do with swimming. Now, here are those cells. These cells are about 1/4 of the cells, and we can expect that these cells do different things. So one of the studies that, let me backtrack there for a second. So you see that there are in fact a lot of cells that are involved in swimming. Now of course the leech does more things than just swimming. For instance, it can also crawl. Here is a typical motor pattern in swimming. This is recorded from a nerve, from a motor neuron that bends, contracts the back like you see in this portion of the swimming. And if you record from the corresponding cell body, you can see that it becomes rhythmically active together with this swim rhythm here. And if you look at the fluorescent trace here, you see that it follows that rhythm very nicely. It is slightly delayed because the particular fluorescent dye that we use here is relatively slow, and the reason is that that little molecule has to travel through the cell membrane. So it's not fast enough to see the individual action potentials. Now this same cell is also active when the leech is crawling. Crawling is a very different rhythm. It instead of undulating, the whole body lengthens and then shortens. And when the body shortens, when that part of the body shortens, that cell fires a lot of action potentials. And notice a slightly different time scale here. This is a minute, this is 10 seconds. So that particular cell depolarizes now at a much slower time scale. And correspondingly, the fluorescent dye follows. When we look at those two things, and we knew from earlier electrophysiology that we should expect that a large number of motor neurons on the dorsal side of the ganglion should be involved in this pattern. And in fact, we would expect that all the cells that are colored green here should be active when the leech contracts during crawling. The red cells should be active when the leech elongates during crawling. Here's the corresponding results during swimming. Some of the leeches of the cells that contract during crawling should also be active when the leech contracts during swimming. For some, the connection is opposite because during crawling, the dorsal and ventral side contract simultaneously, and during swimming, they go in antiphase. Now, it turns out that what we understood from electrophysiology, from individual cells electrophysiology, actually matches the results of imaging very well. All of the cells that you see here colored are pretty much colored the same way based on what Kevin Brakeman and Bill Kirsten actually observed when they did this experiment with the sensitive dyes. But it's interesting to see that there are a number of additional cells that are active. So it turns out that what we thought we knew the central pattern generator. It turns out that there are actually a whole bunch of other cells that are also involved, even though they might not be required for this rhythm. That is, if you send in a current to one of these cells, you cannot influence the rhythm too obviously. Now, in reference to what Jeff told us earlier, it is simply not true that there are areas that are purely sensory or purely motor. And we found that in this study, we can see a beautiful experiment instance of that too. The other side of the leech ganglion has most of the sensory cells. I mentioned to you this particular cell that responds when you hurt the cell, so nociceptor. And on this side, based on the predictions from individual cell electrophysiology, there should be relatively few cells that would be involved in the rhythm. But when they did the experiment, they found that in fact a whole lot of cells are active. Now that nociceptive cell here is not active, but a lot of its friends in that area, none of which we knew had a motor role, is actually involved in this rhythm. So this is a very clear example of why it is important to study whole brains and not just the one little module that you think is important for a particular behavior. Now, that was an example of a motor behavior, right? I'm ultimately interested in understanding sensory behaviors too, and so I'm going to talk to you about a study that we did a couple of years ago about a system where we have everything from sensory to motor, and you can consider it a simple reflex behavior. We have a number of, I should explain the behavior first. A leech doesn't want to get eaten, so if you poke it, you try to nibble at it, it bends away from you. And we understand very well how that works. There are a number of sensory cells that are sensitive to pressure, and each of them is sensitive to pressure in a certain segment of the body wall. So there's a dorsal pressure cell that responds to touch or pressure on this side of the segment. There's a ventral cell that responds to this side, etc. These cells project to local interneurons, which then project to the motor neurons, which project to the musculature. So it's a very, very simple system. Excuse me. And we know the locations of all of these cells in the leech ganglion. We were interested to know whether it really was this simple. So we again used the same imaging using voltage-sensitive dyes, and we found that there are a lot of cells that are active, and many of these were predicted. And when we stimulate the skin periodically, they respond phasically. So here is the time of the stimulus. Many cells respond with depolarization right after, and a number of cells actually respond antiphasically. That means that they respond by hyperpolarizing. They are responsible for relaxing this piece of skin. So if I touch the leech here, it will contract this piece of skin, and simultaneously it relaxes that piece of skin. And here's an example, the purple cells, right over there, which is responsible for that relaxing. So this is all that we would have predicted from the model. Now, we modulated this system. We threw some blocker of hyperpolarization on a gamma blocker called bicuculin thiodide. And what we saw was a dramatic shift. All of these cells that responded at different phases now suddenly became active, became activated by this stimulus. So it turns out that the system wasn't as simple as we thought. It wasn't the case that there were simply these hyperpolarizing negative connections to these cells. In fact, there are also strengthening connections that we just hadn't seen before, but that we can unblock. And as serious neuroscientists, we then washed it out and we saw that the whole thing relaxes. So in particular, this one cell here that was responsible for relaxing here, now not only doesn't relax, but actually activates a lot. It's right here at the arrow. So we conclude that the system wasn't that simple. It's not just the case that the skin projects to this sensory cells, projecting to some neurons, projecting to some motor neurons, projecting to muscles. There's actually a very important inhibitory system there as well. Now, in between sensing and activation is making a decision about what to do. And this system allows us to look at that too. So leeches can both swim and crawl, as I told you earlier. And they need to make a decision whether to swim and crawl. In practice, what they do is if you poke them and they want to get away from you, they will swim if there's plenty of water, and they will crawl if there's not very much water. And they will right. Now the amazing thing about the leech is that you can take the entire system apart and it will still behave. You can literally put this system inside out, peel its gut off, put it back inside in, and it will still happily swim. And trust me, this was done for a real scientific purpose. But what was done in these experiments was we actually just took the entire nervous system out and pinned it down on the Silgard dish. Now, if you take your nervous system out of your body, you're going to not be very happy. You're probably not going to be alive for very long, but the leech can survive in this state for quite a while. And if you electrically poke one part of the nervous system, you can still make this nervous system go through all the motions of swimming and crawling. And in fact, if you choose your stimulus judiciously, you can find the stimulus that will sometimes elicit crawl and sometimes swim. So here are examples of some 10 times they stimulated a nerve here. And in the blue cases, the nervous system performed fictive swimming, which you can see here with this fast swim strokes. And in some cases, it did fictive crawling, which you can see by these much slower fictive crawl steps. And during these experiments, Gavin recorded from the ganglion right over there. And he looked at the activity in all of the neurons. So here's one particular recording from one particular cell in a swim trial. And you can see that the membrane depolarizes periodically like that, and correspondingly the voltage-sensitive dye goes up and down as well. Again, you can't see the individual action potentials there. Of course, the nice thing here is that you can do this not just for that one cell, but for all the cells at once. And if you do that, you get a trace like this. With in the swim trial, you get the cells responding fast, and in the crawl trial, they respond slowly. And you can really hardly see this here, but you can analyze this on the computer. and you can look at the different phases and the different activities. And they were able, by some analysis techniques that I'm not going to go into, to analyze what cells are active in a different way during swimming and crawling. And they found that, in fact, a whole bunch of cells are active in a different way quite early on. And these cells will predict whether the cell will go into ultimately a swimming-like pattern or into a sort of crawling-like pattern. And it turns out that even early on, those trajectories in this analysis space diverge, so that these cells are predictive of making this decision. But there's one particular example here that didn't classify very nicely. And we think that the leech thought of going to swim, but then ultimately decided to crawl instead. So here we get a real insight into the leech's mind. Now, I feel that we still haven't really gotten to the sensory side here. I want to understand the more complex behavior. So all of this was done in the lab of my postdoctoral advisor, Bill Christown. And in the remaining, how many minutes do I have? 5 minutes. My goodness. I'm going to tell you briefly about what I'm doing in my own work right now. So I'm interested in complex sensory behavior, find their prey. Imagine there's a leech in this lake, and it wants to find its prey. That's this guy here. My friend Quentin is inevitably going to make waves when he steps into the lake, and that's what the leech is going to sense. Now, this is a very particular kind of prey because he is actually out to catch a leech. So the leech should pay attention not just to, is this something that I want to go to, but also is this something that I want to go away to. So the waves that he makes are predictive of there's something there. Is this the right kind of prey? Perhaps there are also other waves that are made by wind. So this is a complex task. So we wanted to know how the leech does this. And we moved the leech to the lake into the lab rather than the lab into the lake for technical reasons. And here you can see an example of how the leech finds its prey. If this movie wants to run, I swear it worked just fine this morning. Here we go. Leeches don't like light. If you give them shade, they will hide. And then if you remove the shade, they're temporarily confused. And at the same time, we give them a prey. And in this movie, which you sped up 6 times so that you wouldn't get too bored, the leech decides to crawl toward its prey. You see that it turns around, it sniffs around, it's not so sure, and ultimately it goes there. And when it actually goes there, the poor thing is rather confused because there's nothing there to actually bite. Leeches are very bad at what they do. They find their prey under the best of circumstances, only about 50% of the times in these experiments. Here I'm showing you what happens when we give them waves of different frequencies. They really like frequencies around 8 Hertz, and they really don't like very fast waves. They are not interested in slow waves. But even in the best of circumstances, they only find that prey about 50% of the time. And that may be to do with the fact that they don't really care so much about finding that prey. They really care about not getting eaten in the meantime. The leech, once it does find its prey, will eat 10 times its body mass and be happy for 3 to 6 months. So this was under relatively natural circumstances. Now we wanted to know what sensory systems the leech uses for this. So in our first experiment, we simply switched off the light. And it turned out that leeches do just fine under those circumstances. The curve is maybe slightly shifted to the right and slightly reduced, but it was not even significant. So you might conclude that leeches are not visual. Well, that turned out not to be true. We were able to do the converse experiment too, where we put 2 leech tanks on top of each other. The leech is in the bottom, and we make waves in the top. And the bottom one is on an airtable, so that there is nothing other than visual stimuli that the leech can get. So this is what it looks like in my artist impression. There are visual waves that reach the leech. And under these circumstances, the leeches also can find the prey, but now they are more interested in 2 Hz waves. So there is some non-trivial interaction here between sensory modalities in the normal circumstance. The tuning curve that you get when the leech has all sensors available is not a linear superposition of these two. I don't have time to tell you about how this develops, but I will tell you briefly what we know about the sensory systems. Leeches have a number of simple eyes in their heads, and they have a number of simple eyes in each of their mid-body segments. And co-localized with those are little hairs that can feel vibrations and water movement. So we're going to record from that. And we know in particular one cell, cell 215, right over there, that responds to visual stimuli. When we flash light onto it, it activates, and it is a first-order neuron. It responds very fast. It also responds to moving bars, which is important because if it didn't respond to moving bars, it couldn't be involved in detecting waves. Now, if we put bars going the other way, it responds almost the same, but not quite, and in fact the modulation is a little bit different. I'm going way too fast for you to understand any of this. But I just want to give you a very brief flavor of the things that we can do with our voltage-sensitive dyes here. So we can record from this whole system again using our voltage-sensitive dyes. And sure enough, that cell responds to the voltage-sensitive dyes. And it turns out that there are a number of other cells that do so too. So we're going to just flash light onto the ganglion right now and show you that there are a number of cells that are active. Here comes a little movie of that. You can see the flashes, and you see that cell 215, I believe it's this one, responds to it. No, it's not be that one. Let's do a number of other cells. There are lots of cells that have nothing to do with this behavior. Now, one thing that I really don't like about these voltage-sensitive dyes is their speed. They're extremely slow. We have a voltage-sensitive dye now that's a little bit faster, but we're also exploring another way to do that, to get to single-cell, single-action potential precision, and that's to use multi-electrode arrays. We developed some in our own lab, and this is a recording from a mouse retina using this array. Each little box represents a recording from a single electrode, and this is about 200 milliseconds, and the scale here is, half the scale is 200 microvolts. Here's when we flash some light on it, and you see that many cells in this retina respond to light, which of course is their job. When we recorded with this same system from the leech, we were a little bit less happy. We just put the ganglion on top. We see action potentials, but not quite as many, and we think this is to do with some gunk that we can't quite get rid of, so we're working on it. But eventually we'll be able to record from a lot of cells at very high precision using these arrays. And I'm not going to talk to you about spike shortening. All right, I'll conclude here. I think I'm just about as late now as I was when we started. You can use voltage-sensitive dyes in small grains to record from lots of cells simultaneously, and potentially even from all of them. Multi-electrode arrays may offer a way to record at a much faster temporal resolution, and we hope that we will be able to match those signals to individual cells so that we can get spike trains that correspond to individual cells. Again, in a small grain like this, you have a chance of getting a concise and complete representation that way. We saw that neurons are part of multiple overlapping circuits, and which means that it's not sufficient to record from a small set that you know are involved in the behavior. You really need to look at all the cells at once to see how the whole system responds. And with this technology, I'm confident that not just within my lifetime, but actually within like a decade, we can make very real progress on mapping out entire behavioral circuits and single cellular precision. I want to end by thanking some of the people who did work in my lab. Cindy Harley, John Hare, and Laurens Maljon, who were postdocs that were involved in the last part of the talk. two amazing high school students, Javier Cienfuegos and Matthew Rossi, who did some of the behavioral work, and my technician Andrew Krause, who kept the leeches happy. And I should very much thank our collaborators, Bill Cresta, in whose lab much of the earlier work with the voltage-sensitive dyes was done, Roger Chen, who invented the voltage-sensitive dyes, my collaborators at CalTech, Peter Siegel and Victor Pico, with whom I'm involved in yet another way to interact with neurons, and Marcus Meister, who is working with me right now on the retina. Financial support came from the Broad Foundations and the Burroughs Fund. And if you will permit me one more second, for blatant advertisement, I am looking for graduate students and postdocs to join me in my next adventure, where I will be starting my own new lab at the University of Cincinnati starting in August. And we will be exploring the sensory integration and the ganglion of the leech. So if you know some promising candidates, please tell them to go to my website or come find me today. Thank you very much.
Okay, since there will be a transition, why don't we have the next speaker come up and start to get ready. That gives us a little bit of time for a question or so. Anybody?
Estimate the number of just grossly behavioral kind of configurations of the leech. How many today we're talking about?
You're asking about the number of behavioral configurations of the leash.
Yeah.
Well, we have described, I mean, it always depends on the level of course drainage you want to go to, right? We know leeches can swim. We know they can crawl. They can bend away. They can shorten in response to a stimulus. They can ventilate, keeping their rear sucker down but not actually swimming. We think that that is evolutionarily a precursor to swimming. And they do that if they are in low oxygen conditions. They can scan with their head back and forth to try to sense where waves are coming from. They can do nothing. They can bite, and they can chew through your skin. They can suck blood. They can mate. They can produce cocoons. So those are probably the most major things. I may be leaving out one or two. So there may be a dozen or so major behaviors that have very different motor patterns that this system produces.
So I was surprised you didn't talk about any learning behaviors, you know, like Candela duplicita for the same reason you studied release sometimes a simple animal, but it was all about learning. And have you studied learning in not just habituation, but any other learning attributes of the leash learn?
The question is, does the leech learn? There are a number of synapses in the leech that have been studied and that do show plasticity. Leeches are very bad at learning. We don't think that if you give them this trial 15 times, where they have to find this stimulus, they either learn to do it or they learn not to do it because there's nothing there. But for simplicity, we avoided it. And in all those experiments, we only ever used the leech once at a particular frequency to avoid the possibility that it would figure out that it's being taken on for a ride.
Okay, looks like we may be ready.